Hello everyone. So today we are going to see about finding open reading frame in a DNA or RNA sequence. So as we have completed task 1 and task 2, we are going to do task 3 today. So hope you will find very useful. So let's move on into the session. So our previous task, what we have learned is we have learned how to download a biological sequence and we have done the translation of our DNA sequence into amino acids. So in this task, we are going to find the open reading frame so that we can understand a particular protein coding potential of our particular gene. So as we begin, first we have to know what is ORF. ORF is a open reading frame in a region of particular DNA sequence. If there is a DNA sequence, there is an ORF region. So that can be translated into protein. So it is like a start codon and stop codon. Start codon is a starting point. Usually it is denoted as a ATG. Okay, and in the end of the stop codon it is always denoted as a TAA or TAG or TGA. So we have to, in between we have, there is a between between start and stop codon there is a series of codon that encodes to the particular amino acid so that it can make up the particular protein. So by finding ORF in a particular DNA or RNA, we can identify the potential protein coding regions of the particular gene. So we can also distinguish between protein coding region and non-protein coding region, that is exons and introns also. So next step is we are going to do the practical. So first up, we are going to open the sequence that we have downloaded from the task 1. Okay, we are still continuing that sequence, that DNA sequence file. Then we are going to open the BioEdit software. Then we are going to load the sequence. So after loading the sequence, we are going to choose the ORF finder tool in BioEdit software. So for that, we have to go to sequence menu, and then there will be a menu called nucleic acid. Select that, and there will be a drop down menu which is called find ORF from a list of positions. Okay, so there will be a drop down menu, and we have to choose the particular parameter. So in this thing, so we are going to find the ORF from a list of question in the dialog box. So there will be a default setting that already been incorporated in in that software. So the particular sequence, if the sequence whole length is 1 to 629, it will carry 6 frame translation, that is both forward and reverse direction, to find the region which is starting from ATG and which is ending from the stop codon. Okay, in this thing we have to enter the maximum ORF size, minimum ORF size, and our start codon and our stop codon. So next thing is we are going to find the ORF. For that, we have to translate the button to find ORF in your sequence. So once the ORF is completed or translated, it is always displayed in a new window. So along with the start and stop position of the length and the protein translation too. So we have chosen the parameter, we have translated the ORF, and the new window will be displayed. And next thing is, by finding what we are concluding is, by finding ORF in a DNA or RNA sequence, we can identify the protein coding region and non-protein coding region to the particular sequence. So let's move on to the practical session. So here is our BioEdit sequence alignment editor. So what we have to do is, we have to go to file, we have to go to open, and we have to change the file type which we are going to upload. So whether it's FASTA file, changes to FASTA file. Whether it is gene bank file, changes to gene bank file. Whether it is BioEdit, so we can change it. Go click on to sequence and give open. So your sequence has been loaded. Now what we have to do? The next step is, we have to go to sequence. Click the icon for sequence. Go to nucleic acid and click on to find ORF from the list of positions. Okay, just click. So before doing this step, we have to select our sequence. So how to select your sequence? Just go to your, click to the sequence ID and go to nucleic acid and go to find ORFs from a list of positions. So we have to enter the position. So what is our position? So for example, 1 to, we have to type this 1 to 6 to 9. Okay. Then you have to give the, press the translate. So it will open to a new page and you can see the frames. From the stop codon to the start codon. I hope this practical session will be useful for you. Please do try it on yourself and share this knowledge to your friends. And please do complete this task. So as bioinformaticians or researchers, this can be used to identify the potential drug target. If you have any drug targets as ORF, so it could able to encode some essential proteins to a particular pathogen which you are targeting by the drug. So we have to understand how that particular gene is evolved. So what changes happens in ORF which can lead to the gain or loss of the particular protein function. So that is mandatory. So I hope this practical and this PPT guide have shown you how to find the protein reading frames in a particular DNA sequence or RNA sequence using BioEdit. So you will get skillful knowledge how to identify particular protein coding region, and you can always distinguish between the coding region and non-coding region. So now it is your turn to do the same practical. So if you completed your task 3, let us know in the comment section. Please do like and share it so that others can learn it along with you. So thank you. Hope you enjoy this practical session.